And the next talk is by Yurii Aulchenko, Institute of Cytology and Genetics in Novosibirsk, about quantitative genetics of human protein and glycosylation. It's written there.
Right. Okay, sorry. Need to get used to. No, it's the current. Okay. Well, I'm going to present our project on, it's a big project on finding genes which regulate variation of protein and glycosylation in human populations. And I would like to start with reminding you that protein glycosylation is one of the most abundant post-translational modifications. It consists of adding an oligosaccharide, a glycan, to specific glycosylation site to the protein backbone. And as such, it changes physical, chemical, and biological properties of proteins. So, but most of the time, I'm going to speak not about, protein glycosylation in general. I'm going to speak about glycosylation of very specific but very important protein, immunoglobulin G. And all of you know that this is a central molecule in adaptive immunity, and it consists of two domains. The upper domain on this picture is antigen-binding domain, right? And then the lower domain is the crystallizable fragment, which is responsible for such effector function as antibody-dependent cellular cytotoxicity, ADCC for short, and also for complement activation. Right, and here, these small brown structures are glycans which attach to a specific site of IgG, to the backbone of IgG. And then you may say, actually, in my old immunology textbook, there was this schematics of IgG, and there was nothing about glycosylation of IgG. So is that at all important? Well, that's very unfortunate, but it's actually very, very important. It's very unfortunate it is not in some older textbooks. Because if you remove this glycan from IgG, it totally loses its effector function. It cannot induce ADCC anymore. So it is very important. Right, here is an example. This is rituximab. This is monoclonal antibody, which is on the list of WHO essential medications. And the original formulation, it carries the glycans like this, right? And then you can see how by increasing the concentration of it's very interesting. I don't know how it works. But this red line is showing you how by increasing the concentration of this antibody, you induce antibody-dependent cellular cytotoxicity, right? Response in NK cells. And then if you change glycosylation, right, essentially you fix it and you remove this core fucose, this red triangle towards the right side, right? You can induce the effect at much smaller concentration and actually can induce much bigger effect, right? And, well, just imagine this is the difference of 3 orders of magnitudes. So think of this. It's as if with the original formulation, to treat certain number of patients, you need to produce 1 kilogram of your monoclonal antibodies. While with changing glycosylation, essentially just removing one little, molecule, you are going to need, instead of 1 kilogram, you can produce 1 gram, right? And you can treat the same number of patients. So I hope I convince you that glycosylation of IgG in particular, but also glycosylation as general post-translational modification is very important. And now let's think, like, if you take a drop of your blood, there is a lot of IgG there. What's the spectrum of glycoforms? What type of glycans do you observe on IgG of regular person, healthy person? Okay, we did that. We do it all the time. And, well, basically, this is chromatogram which shows the distribution of different IgG glycoforms in a normal, healthy person. So you see about 2 dozens of major glycoforms are present in the blood of any of you, basically. All right. This distribution, though, it's not static. So if I compare the distribution of glycans, IgG glycoforms between you, I will see differences. Moreover, as I age, this distribution will systematically change. And also, if you look into certain disease states, especially autoimmune and inflammatory, there are systematic changes to this profile. Okay, so it does look like a complex trait, actually, right? For you, for me, like for a quantitative geneticist, I would say, okay, what's heritability of the height of these peaks, right? And if you study this, you will figure out that heritability is rather typical for quantitative traits, like 30 to 70% on average. So this protein N-glycosylation, IgG N-glycosylation, is a complex trait. And we had a paper published which saying just that. Now, there is this very useful notion when you think about complex traits, the notion of core genes and peripheral genes, right? So there is this idea that when you talk about complex traits, you can think of 2 sets of genes, one related to the core pathway. So basically, this is a set of genes which exhibits direct effects on the phenotype of interest, while there is also huge regulatory periphery. So there is a big set of genes which exhibit their effects through regulating the core genes, right? So this is, I think, very handy, very important notion. Core set of genes and periphery which regulates this core set. And so if you think of complex trait, this regulatory periphery is something which allows your trait, your phenotype, to react to external and internal stimuli, right? That's quite important. So let's think what we know about protein glycosylation. What do we know about core pathways and about the regulatory periphery? And, well, actually, that's a beautiful trait to work on because we know a lot about at least one of the core pathways, because biochemists, cell biologists did a brilliant work figuring out all these chains of reaction which convert fucosyltransferases, which convert one glycans to other glycans, in which compartments it happens, and so on and so forth. So a really great work, and we have very well-characterized core pathway. So, but what about the regulators, this huge regulatory periphery, which we should observe in any complex trait? What do we know about that in humans? Because, you know, the picture should look like this. There should be a lot of regulation going on. And specifically for humans in vivo, we know very little, close to nothing about how this thing is being regulated. Okay, so this brings us to like grand goal of our big project. We want to figure out what is the network of genes which regulate these core pathways, and maybe also figure out some other core pathways involved. So how we do that? Well, being classical geneticists, we are following the experiments of nature. So we try to relate the naturally occurring germline standing variation with the variation of phenotype of interest. So we can call it positional cloning. That's something which was done for many tens of years by now. So we start with running genome-wide association studies. We find loci associated with the phenotypes of interest. Then we do a lot of bioinformatics works, and I would like to thank Shamil for describing some of the methods which are being used. So we use pretty much the same methods. It's rather standard. I will not go deep into that. And then finally, you definitely want, when you worked out some hypothesis, when you postulated mechanism of action of some locus, and you believe it may be true, you want to go to the lab and test if it works or not, right? So this is this three-step approach of positional cloning. Okay, very good. So we've run a genome-wide association study, studying the profiles of glycosylation in about 10,000 people. And this is a bit unusual Manhattan plot, which shows on the lower part is what was known before our last study, and on the upper part, the loci we found in the new wave. And on the top of each locus, there's these tags. These are the genes which we think are potentially the causative genes in the locus. Okay, so what we can see here is I think rather interesting, because immediately if you're a glycobiologist, you can see there are elements of core pathway. So here are these ST6GAL1 and other genes marked in orange, right? These are all the elements of the core process. This is pretty cool. We can immediately see them. And here I must say something. Well, Shamil, your talk was very interesting, but here we see something very different. So for 3 of these 5, we can do good colocalization when we use CD19+, so B-cell lineage cells. And for the 4th, there is a structural variant. And for number 5, I'm sure that, well, I'm not sure, but I kind of foresee that when we finally get hold on plasma cell expression data, because we are now using proxy CD19+ cell lines, cells. So when we get hold on the data for plasma cells where indeed this glycosylation occurs instead of proxy, potentially all 5 will be nicely explained. So I think this happens because for this system, we really know a lot where it happens, how it happens, and probably for complex disease, it's way less straightforward, right? But here, the picture is, I think it's very, very nice trait to work on, very, you know, rewarding in a way, right? So, good. So these are core pathways. But I started with saying, like, we want to figure out what is regulatory periphery, what is regulating this core pathway. And here we have a number of genes marked in green, which are either transcription factors or something to do with transcriptional regulation. Right, so we have a set of core pathway, these orange genes. We have a set of green genes. Can we relate green genes to orange genes, right? So can we say which green label regulates which orange label? And then here there is another very nice feature of the trait we are working on, because glycosylation, you've seen this profile, right? It's multidimensional. So we thought that if a green gene regulates orange gene, then the footprint, its effect on the glycomic space should be the same. So what we did, we built this proximity network, right? So where nodes are clustered based on the similarity of effects onto glycospace. And well, let's have a look at this specific, 3 nodes. Here we have FAT8. This is fucosyltransferase. It does core fucosylation. Very important, very good. And then we have Icarus1 in one node and Icarus3 in other node. So both are known important transcriptional factors, specifically in B-cell lineages. Right. Okay, so our hypothesis is that Icarus1 and 3 somehow potentially may be regulating, so these are peripheral genes, and they may be regulating specifically FAT8 from the core. Okay, well, we can go to the lab and test it. So that's what we did. We went to LCL and knocked down expression of Icarus1. And then what we've seen is that Icarus3 expression went down, and then FAT8, our core gene expression, went up. And actually, we can follow it through to the final readout. The degree of core fucosylation of IgGs produced by our cell line was also going up. Right. I think this is like nice coherent story where we start with, with GWAS, and we go all the way through, and we figure out potential mechanism of action and confirm that in the lab. So in principle, we think that our first iteration over this story is over, and we will try to expand. So at the next stage, quantitative geneticists, they're terrible people. They are power hungry. So we always want more. So next stage, we want to put this story on steroids. Instead of 10,000 samples, which we now used in GWAS, we want at least 20, better 30. We want more omics data, better matching to specificity of the trait we are studying. We are really I dream to have plasma cells, as many as possible. We want to do more proteins because we've been looking only in IgG, all right? And actually, we are not been looking very precisely because like there are 4 isoforms and there is actually not only FC but AFB glycosylation. So we want to go more precise, but we want also to go broader. Right now we are also doing transferrin, and Sodbo in the next talk is going to talk about our other effort. Right. What is important to realize is that we use genetic methods, so we are not reconstructing full networks, right? We only see nodes where there is this variation which leads to phenotypic variation, right? So this is important to notice. If you want full network, you would need to do a more system biology type of approach. Right. So this is more or less it. We think that by exploring this regulatory periphery, we will see how the glycosylation relates to, disease, to the aging process, because we don't think it's sitting in the core process. It's more like on the periphery. And that is also one of our grand aims. And I should say that this work would have been impossible without, this large group of bright young people working with me. And I also would like to thank our collaborators from Edinburgh and Zagreb, and you for being here and listening to this. I'm open for questions.
Okay, there is a question. Sorry, question from Vanya Kulakovsky. Did you check if transcription factor to target gene links in the network are additionally supported by manually curated or high throughput data?
Specifically for this little triangle, we did that. I think we didn't do that systematically enough. This is something for the next round because, well, that was a long project. I think the short answer is yes, but we definitely didn't do that enough. So we want to do that for the next round more thoroughly.
The short answer is yes, but not quite. Okay.
Precisely.
Any questions from the audience?
Okay, thank you very much. I was wondering whether there are any strong genetic correlations between traits in the human phenome, let's say, and the protein glycosylation. Having a look at that.
So if I understand you correctly, you're asking if there are genetic correlation between the glycosylation traits and other traits, right? Okay, that's a very good question. That's why we are power hungry, because with 10,000 people studied, we don't have power enough to do much of the genetic correlation analysis, at least using the summary statistic-based methods like LD score regression and others. I think that's the short answer. We do see something, but 2/3 of the time the algorithm fails just because we don't have enough power. I think there is a poster which talks about Mendelian randomization study between a set of inflammatory and autoimmune disease and glycan genetics, so somewhere online. So that's
Yurii,
kind of step into that direction also.
Yurii, I have a general comment about the state of the community, I guess, because this is the first talk I observed ever which discusses the omnigenic model as the reality. Right? We all like the model, but it was sort of semi-controversial in the semi-controversial space. So the first time I hear you stating it multiple times as reality. So I don't know if you'd like to respond, but it's a general observation.
I'd like to respond definitely. I even didn't call it omnigenic, model. I didn't, in a way, give a credit. But here the important thing is that I think that in general the omnigenic model is controversial, and also when we try to make a precise formulation of it to put it in test, because well that's what we've been trying to do, we find that it's laid down in a way which is very hard, it's slippery, right? But I think that the part which I was mentioning, I think most of the people would agree that for complex traits, you do expect set of genes which work by regulating other genes. Would you agree? And that's the only thing I told. There is these core genes which exhibit direct effect and periphery which regulate the other set. I think that's not very controversial. I think that's very, I shouldn't say straightforward, but I think it's very valuable notion. I was also avoiding saying hypothesis or theory, but I think this way of thinking is very, very useful for us at least, where we have this very clear core pathway. Right? I think it's very fitting to our specific field.
Okay, and let's continue. Thanks a lot.